Israel. I'm a PhD student at ARO Volcani Center in the Faculty of Agriculture, UG, and my research is focused on the conservation and characterization of wheat landraces from Israel and Palestine. Professor Nana Meir gave me this wonderful opportunity to share with you my work on the conservation of landraces from Israel and Palestine and their relevance to production and semi-arid regions based on two-season field study. This project is a collaborative effort including several research institutes in Israel. Since Israel is currently under lockdown due to pandemic as well as the obvious time gap, I'm pre-recording my talk, and I hope that I will have the opportunity to meet you during the online course. Israel and Palestine and vicinity at the southern tip of the Fertile Crescent is a center of wheat genetic diversity. During the last 10,000 years, it has also hosted a continuum of wheat crop cultivation in a wide ecogeographical range of environments. The value lies in the wide range of variability, specific adaptation in the regions of growth and distinct culinary and cultural uses. The challenges in increasing food supply and the selection of elite crop varieties has resulted in the loss of much of the genetic diversity. Landraces are of great potential for broadening the narrow genetic variability. Israel at present time is an extreme example. We understand that the transition from traditional to modern cultivars resulted in an almost complete loss of this unique resource. A number of collections expeditions held in the 20th century of wheat landraces in Israel were conserved ex situ in gene banks in private collections, and some were in real danger of extinction. The research goals are restoration, the Israeli-Palestinian landrace IPLR collection, conserve the IPLR collection, characterize the genetic and phenotypic diversity of the IPLR, compare the agronomic and multi-site field study, examine the feasibility of cultivating landraces on the local market. In this talk, I will focus on these two goals, and last one is a genome-wide association mapping of agronomic and yield-related traits. During two seasons, we carried out a field experiment comparing agronomic traits of diverse panel of spring wheat landraces and modern cultivars. Among the landraces group, we chose 7 durum lines, 6 spread wheat lines, and a mixture of each of them as appears at the table. Some of them have traditional names such as Horani, Nursi, Gaza, Jaljuli, and so on. Modern cultivars, both durum and bred wheat, grow today in Israel, were chosen as a control. The experiment was designed under 5 environmental conditions: semi-arid under 2 water treatment with and without supplementary irrigation. This is the Gilat size for 2 seasons. And under Mediterranean climate condition, this is the Beit-Dagan experiment. In Israel, we sow spring wheat in the winter, around mid-November, and harvest around the end of May. These are the sites where we set up the experiments. Monthly temperature range from 40 to 110 Fahrenheit in all fields. In Gilat, a semi-arid climate with precipitation of only 260 to 360 millimeter rainfall during 2 seasons, and in Beit-Dagan, a Mediterranean climate with rainfall of more than 700 millimeters in one single year of the experiment. The selected landraces in the field study form a tiny portion of the entire IPLR collection. The construction of the IPLR started with locating and identifying in GeneBank's database of any wheat accession that was cultivated in our region. This was done based on sample description, typical traditional names, or sometimes specific collection sites. By that, we could construct the collection from 18 different donors. And interestingly, 2 of them were Israeli private collections which were poorly preserved and were on the verge of extinction. This is the Matatia in the IGB and the NMF collection. The collections contains today more than 900 accessions, where about 2/3 of them are durum. The entire collection was propagated and characterized in a common garden experiment along with a control panel, 56 modern cultivars that represent Israeli wheat varieties grown for the last 50 years on farmer field. In collaboration with CIMIT Institute in El Batan, Mexico, we genotyped with 26K markers the entire IPLR collection using DartSeq platform. The IPLR collection expressed a wide diversity, both in tetraploid and hexaploid subcollections in comparison to the modern cultivars control marked in blue dots. The diversity is also expressed in spike morphology, such as size, density, shape, and color. These are the durum example. and these are the bread wheat example. Back to the field, when we analyze the field data, we see a clear separation between modern cultivars marked in solid cells and land races in all 5 environments. The major vectors driving this agronomic variability are the green revolution traits, such as grain yield, harvest index, 1000 kernel weight, gluten index, while the land races characterize in higher plant, higher protein due to lower yield, and higher tillering and diverse heading period. In a drone image that was taken at the end of the first season in 2018-19 in Gilat, we observed severe lodging mainly in land races, for example, the Juljuli. In some cases, lodging started in early stage, with extreme effect during or after heading. Some land races, although characterized by high plant stature, did not lodge, which was positively surprising. In the left image, we compare grain yield differences between 2 growing seasons in Gilat. Each exhibit moderate and extreme water deficit. Interestingly, the land races manifested greater stability in comparison to modern cultivar during 2 seasons in Gilat. On the right image, earliness seemed to be a crucial factor for promoting yield among land races. One of the research outputs is the selection of 5 land races with superior performance across growing conditions. Integrating yield, earliness, non-lodging, and quality parameters that we are about to see, we chose 5 land races for further propagation, 2 bread wheat and 3 durum wheat, Lubnani Kisra, Dirala, and durum of Hitia Soada, Gaza, and A238. So in addition to the agronomical characterization, we also looked on the baking characteristics and quality attributes of white and wholemeal flour. Principal component analysis of the IPLR core set based on quality and baking parameters points a genotyped mean for white, these are the yellow circles, and wholemeal, these are the red triangles samples. Grains were harvested and evaluated in the bread laboratory in Gilat. Interestingly, elite cultivar Gadish samples that are marked in blue dotted circle show slight difference between white and wholemeal flour. On the other hand, the sample of Juljuli, a bread wheat landrace marked in green dotted circle, show high discrimination between white and wholemeal flour. This emphasizes the potential role of landraces bran on quality parameters. At the current stage, we constructed together artisanal bakers in an expert panel to examine their feasibility as varieties for the Israeli artisanal market. This season, we collaborated with artisanal bakers who were passionate in growing their own wheat. We provided them seeds from our field experiment. 2 of them are in the north part of Israel, as you can see on the map. In the south, a farmer decided not to sow the landrace Gaza due to low precipitation at the beginning of the season. So, the conclusions so far. The IPLR collection is a highly diverse and exotic germplasm in comparison to the narrow genetic-based germplasm of modern cultivars currently available for farmers and breeders. Field data highlight the strong imprint of the green revolution in reducing plant height, raising harvest index yield, and also promoting earliness. Under semi-arid conditions, land races exhibit yield stability, although this stability might be associated with low productivity. 5 land races have shown promising agronomic potential and will be further examined for their artisanal potential. Adjusting the right field management protocol should be considered before those are used by local farmers. I want to express my deepest gratitude for my thesis supervisors, Dr. Roi Ben-David and Professor Shahal Abbo, to Dr. David Bonfil for his collaboration and endless help in the field experiment, to all the researchers listed here who formed part of this amazing project, to the lab members of Roi Ben-David Lab who held to carry out the field data, and I want to thank my family that are with me at home, making it possible to participate in this wonderful course. Thank you so much for your attention. Bye-bye.